Hi students, good evening. Welcome to the rapid revision round and a very interesting topic with transport in plants. Okay. So I welcome everybody for this interesting session. Okay. So let us now start today's topic with the interesting and interactive and informative session on the rapid revision on the transport in plants. Okay. So in today's class, okay, we are going to see about the transport in plants. Now when it comes to this topic, mostly I have seen nearly 8 to 12 marks of questions have been distributed every year from this topic, like in the problem, like in the form of some problems, or they might ask some pictures or match the option. Like that the questions will come from this chapter. So from the NEET perspective, this is an important chapter that we need to know. So I would like to say that it is very important. So hi students, Thirisha, Murugan. Okay, we will start with today's interesting, interactive, informative, rapid revision on the transport in plants. Yeah, we are going to the first question here. Okay. Okay students, we will start now. So look at this. Okay. So here in this they have given, based on the figure given below, which of the following statement is not correct? Okay, so they have given a picture. So these are the solute molecules. The blue one is the solvent, the water. This is a semi-permeable membrane. So there is A and B, a partition made by a semi-permeable membrane. So what are the options they are given? They are asking something which is not correct. Movement of solvent molecules will take place from chamber A to B. Movement of solute will take place from A to B. Presence of semi-permeable membrane is a prerequisite for this process to occur. The direction and rate of osmosis depends on both the pressure gradient and concentration gradient. Now students, you should know here, like we are talking here about osmosis, yes, and here what is that? There is the movement of only the water molecule from a region of lower solute concentration to a region of higher solute concentration, and the semi-permeable membrane is mandatory and it depends on various gradients also. So which is not correct? The movement of solute is not correct, which will not take place from A to B. So I see Jivita, Thirisha, Akshaya, everybody, like they have started their usual way of bombarding the answers before I put forth the question. Excellent students. Okay. So now we will see about the next question here. When a molecule moves across a cell membrane independent of other molecules, the process is called option A, uniport, B is Simport, C is antiport, and D is facilitated diffusion. So what does it mean? So here, so imagine there is a membrane, right? So you are having a membrane and they are saying there is only a movement of a independent molecule across a membrane. So what you call that as? Okay. So we will look into these options. Now when I say Simport, okay, Simport is what actually? In Simport, what is going to be? It will be something same like your co-transport, like where two molecules can sit together and there will be some configuration change and they will be pushed to the inside or outside of the cell. So where two molecules will move. What about in facilitated diffusion? Okay, in facilitated diffusion you need a carrier protein, right, and the molecule will get stuck up with it and there will be a conformational change for it to push in. But here that ATP will not be needed, whereas here the ATP is required. What about in antiport? In antiport it is something reverse. If one molecule is on the outside, the other molecule will be inside. So they will move in opposite direction. If one is moving out, the other will be moving inside. Like mostly it happens in our kidney, renal tubules, right? So there and all we can come across with antiport. Whereas Simport, that co-transport or the facilitated diffusion, you can absorb in the, you can see that in our gastrointestinal tract. But what about the uniport? It is a single molecule that is moving. Depending on the concentration gradient, there will be just movement of only one molecule across the cell. So that we call it as uniport. So that should be the answer. Okay, very good students, like you have given the answer. Now here we will come to the third question. When the conditions are dry, a grass leaf curls inward to minimize the water loss due to the presence of thick cuticle, large xylem cavities, parallel venation, bulliform cells. So what should be your option? So we'll see what are bulliform cells. These bulliform cells, actually you see they are sort of the epidermal cells, right? So what usually when there is enough water, whenever there is enough water, what will happen? They will just absorb the water, okay, and they will become turgid, okay, so that the leaves, whatever they are getting exposed to sunlight, and so the water loss will occur. But when the bulliform cells, that is when conditions like when there is very dry, when there is water scarcity, what will happen? The bulliform cells, they become more flaccid because of what? The water loss, okay, and that is a reason for the curling up of the grass leaf to minimize the water loss. So these two things will not fit into the option. So students, you will make mistake between the thick cuticle and the bulliform. Now I am asking you about the grass and the thick. This is a very adaptative of a xerophytic sort of adaptation. So we don't take this also into consideration. But bulliform cells with grass, so what is that? They become flaccid because there is water loss. So immediately they will curl up inward so that they will minimize the water loss. So this should be the answer. So what is the answer? It is the bulliform cells, like as you observe. So what they will do? They will become more flaccid whenever, but usually they are very turgid in sufficient water condition. But when they are in dry condition, so what will happen? When they are exposed like that, they will become more flaccid and the water loss will be there. So that results up in curling up of what you call the grass leaf to inward, so that will minimize the water loss. So the answer is bulliform cells. So I see students giving up the answer. Excellent, ma. We'll move on to the next. Yes. Now coming here in land plants, okay, that is our terrestrial plants, the guard cells differ from other epidermal cells in having chloroplast, cytoskeleton, mitochondria and endoplasmic reticulum. Now you should see that how are they different. Like you see the guard cells, how they are different from any other epidermal cell that is present in the plant cell wall. Now when you look into the cytoskeleton, that is present, it is characteristic of all the living cell which is there, even in bacteria we do see. So this we will not take into consideration. But what about mitochondria and endoplasmic reticulum? Even these are also there in all the eukaryote cells. So the difference that we are going to see is the chloroplast, because here the guard cells different from other epidermal cells is because of the presence of the chloroplast, because they can manufacture their own food. So that is where they are different. So you can see so these are the guard cells, okay, and then we'll have the subsidiary or the supporting cell, and you can see that small pore like opening which we call it as the stomata. So these guard cells are the one which will regulate the opening and closing of the stomata. So what you see, those green ones here, they are the chloroplast. So what should be? So the guard cells are quite different from any other cells of the epidermis by having the presence of chloroplast. So the chloroplast is the one, okay, so that is also very important. Why they are important? Because they are important for the activity, like probably for regulating the opening and closing of the stomata. So that is where they are different. So that should be your option. So I see Sri, Sirisha, Murugan, Jivita, Akshaya, everybody gave the right answer for this question. Excellent, ma. Good. Okay, excellent. Now when it comes to these questions, like every day as I take the rapid session, I am emphasizing this one thing pertaining with biology, that students, when you read a statement, you are given 4 options. Of that 4 option, which is the nearest possible right or the correct option you are choosing. Why not the other 3 options you have to derive? So don't think that in NEET physics, chemistry will have the derivation. Even in biology, you need to derive. Derive what? Derive the nearest possible answer. So why you are not choosing the any other 3 option or choosing the one nearest option? That derivation part, as you make it out, you will avoid errors and you can score marks with flying colors. Okay. So see here, the rate of transpiration is measured by Genong's potometer, porometer, oxynometer and respirometer. So there are a lot of things given. So we know what is transpiration, right? That water loss, that is right present in the leaves, right? So how is that rate of transpiration is measured? So some amounts of water they are getting lost, okay, through the leaves. So that we call it as transpiration. That creates a what you call a suctional pull, and that in turn is offering the water absorption through the root. All that we know. So how are we going to measure this transpiration? So the transpiration here, we are going to measure the rate of transpiration is measured by using what is called the Genong's potometer. So that is what do you mean by porometer? Here we will measure the relative size of the stomata. So students, what they will think, something, that is what they wanted to confuse. So here it is something to do with stomata. So don't ever choose this option. What about oxynometer? That will measure the plant growth. We have Pfeffer's oxynometer. I hope you would have studied about that also. So here it will measure the plant growth. What about respirometer? It will measure the rate of respiration. So you have to be very clear. So what they have asked here in the exam. So these are the derivations that I wanted my students to make it up either in the mind or in somewhere in a worksheet, so that you will know what they are asking for. So here it is the rate of transpiration. So what should be your option? It is the Genong's potometer. So what you observe here in this picture, this is how they will find out. Okay, so you have that. So how much of air bubbles? That is where we have a volume scale also, and that is how they measure it. So this is what we call it as the Genong's potometer. Okay students, you are able to follow. So very good students, you have given the right answer. Yes. Here comes another sum. A cell with DPD. Okay, so that is DPD is what? The difference that is between the osmotic pressure as well as the turgor pressure is 5. That is surrounded by A, B, C and D cell having OP and TP respectively of 5 and 4, 7 and 5, 3 and 2, 0 and 0. From which cell it will withdraw the water faster. Now I told you that diffusion pressure deficit is nothing but the difference between that osmotic pressure with the turgor pressure. Now what they have given with cell A? They have given it is 5 and 4. So how much it should be? 1. What about with B? It is 7, right? What it must be with the cell C? 3 and 2, right? So what it should be with 0. So what should be the option from which that withdrawal will be faster? So the withdrawal, they will say that it is more in A. So what you have given the answer is right students. The answer is actually A. Excellent. Okay, good. So we will go to the next question. Here we are going to match some of the important interesting things. So they have given column 1 as well as column 2. You are going to select the correct option here. Okay. So what is the correct option? We will see. So they have given leaves, seeds, roots, aspirin, plasmolysed cell, and in column 2 they have given some of the functions of these structures: anti-transparent, transpiration, negative osmotic potential, imbibition and absorption. So you should know like what option that it should fit. Okay. So let us start working on it. So once when we see leaves, immediately where we will jump, we will think of the transpiration. Yes, the leaves that has got the capability, like where the water will get lost from the water from the leaf surface. What about the seeds? Yes, there is a process of what we call it as the imbibition, where it will get just imbibe the materials into it. What about the root? The roots are meant for what? The absorption of minerals and water from the soil. What about this aspirin? So when it comes to aspirin, what should be your answer? So here the answer for this aspirin is what? It is nothing else then it is going to create, it is going to create the anti-transparent effect. So aspirin, it has got an anti-transparent. So what do you mean by anti-transparent? That will not allow the transpiration to take place. And what about the plasmolysed cells? The plasmolysed cell, okay, it will exert a negative osmotic potential. So the nearest possible is leaves. We all know that is with transpiration. See children, how you have to cut down to the answer. See, it is very direct, straightforward question that leaves with transpiration. So it should not be these two. So you are going to do along with it. Next, coming to the seeds is imbibition, right? When it comes to roots, yes, absorption. Okay, so the roots, it is absorption. Okay, so that fine. What about when it comes to aspirin? So here only it makes, but you will be very much thorough with the plasmolysed cell which will have the negative. So this cannot be. So what it should be? The negative osmotic potential. So that should be your answer. Okay, so the answer is what? It is what we have marked. Okay students? Yes, we will go for the next question. The pathway of water movement from soil to xylem. Yes, I see Jeevita, Sirisha, Balamurugan. Yes, very good. Even Murugan, everybody are giving the right answer. Student, please do interact. Okay, very good. I see you all posting the answers. Excellent, ma. So coming to the path of water movement from soil to xylem. So this will be really very nice. See how it is going to move. So now you should know that they have given all these options, like it is right from metaxylem to root hair, or cortex to metaxylem, or soil to metaxylem, or pericycle to metaxylem. First we should understand that whenever we talk about that water absorption, so these are the key points. Yes, the water are absorbed by the root hairs. Okay, so what is happening? It will make the internal medium hypotonic. Okay, so the absorption of water will become more passive. It will go along from a region of higher concentration, okay, to lower concentration. So that is how the water is going to move without any energy expenditure, say like a facilitated diffusion. Mainly we have what are the water channels, we call that as aquaporins. Only through this the water movement will happen through. So the root hairs, okay, they are going to absorb it from the soil. So how the movement would be? So it should be soil, the root hair, then it goes to the cortex. Yes students, you are all right. Then we have the endodermis, then the pericycle, the protoxylem, the metaxylem. So this is how the minerals and water, the solutes, they have been taken up into the xylem. Okay, so this is the movement. So think of these root hairs and how is that internal medium is hypotonic. So there will be a movement of from a region of higher concentration to lower concentration. And we have a specialized water channels, what we call it as aquaporins, and through that only the water molecules are getting absorbed. So this is an other way. So soil, root hair. So it cannot be from inside to outside, or it cannot be from cortex, root hair and all. So the first the root hair should absorb. Same way not the pericycle which is inner. So you have to find out which is the odd one and choose it. Okay. Excellent students, you have given the right answer. Here comes another interesting question. See, a man supplied excess fertilizer. So this usually happens with our farmers, okay, where they will come with all these issues. So what has happened here with this farmer? He has said that there was excess fertilizer and watered the grass well. So he has given more of fertilizer and he has given enough water. After some time the leaf turned brown. All the leaves become brown in color. Why? Because exosmosis occurred in root and the plant dies. Fertilizers were drained in the lower layer of soil. It decreases photosynthesis due to water logging of soil. So what should be your answer? I am waiting for your answer students. Please do post it in the chat box. Now when you see the plant, okay, so when you put excess of fertilizer, what will happen? They will be actually getting absorbed more in the soil, right? So you have the concentration of the soil solution, what it will be more hypertonic, okay, when compared with the root solution. Okay, so what will happen? So because why it is becoming more hypertonic? Because he has added that excess fertilizer. So because they have added excess fertilizer, it has become more hypertonic when compared with the root solution. So what will happen? There will be exosmosis. What do you mean by exosmosis? So there will be movement of the water from the region of what you call, from the lower solute concentration to higher solute concentration. So there will be net movement of water. So the exosmosis will occur from the roots. So when all that comes out, what will happen? The viability of the plant will be under the check. So there will be shrinkage. So what will happen to those roots? So there will be shrinkage, okay, of protoplasm, and there will be what is called the plasmolysis. That's the reason why it happens. Okay, so that is the reason. So there will be what is called the exosmosis. So always because the solute concentration is more outside than inside the root. So definitely we saw in the first slide itself in osmosis. So there will be exosmosis, all the water will come. So there will be shrinkage, and so the plant will die. So that is the answer. Okay, so you are able to understand. Yes students, very good. I see here a lot of people have given, because this is a question where you have to apply your logic. See, in reading biology or physiology or medicine, always we follow this, only logic and common sense. So if you apply your logic, very easy this answer is. Right students? Very good. I see Tirisha, Akshaya, Murugan, Jeevita, then Bala Murugan. Excellent. You people are just giving excellent answers from the beginning. Very good show. Very good. I really say you are doing a good job. Okay. So this is how you have to understand what is meant, and you have to make the answers. Okay students? Very good. Okay. Now here comes the, they have given a lot of structures in column 1 and functions in column 2. We are going to match the structure and function, which is going to be a very easy job children. So look here, stomata, hyadothode, root hair, xylem vessel. Here they have given ascent of sap, transpiration, guttation, absorption of water. So how are we going to match? So we know very basic things, like when you are very much clear. Okay. So we can very well make that. So what are we going to make here? Here we are going to see the stomata. So what about in stomata? We know that stomata is going to help in what? The transpiration. What about the root hair? The root hair, yes, absorption of water. These are simple questions, like where we have been learning these concepts for years together, right? Right from our KG classes. We know this, stomata with this. What about the hyadothode? That is something to do with guttation. So what is guttation? Like while we know that transpiration, it relies on the stomata, guttation is with the hyadothode. So there will be actually exudation of drops, those of the xylem sap on the leaf margin. Like when you see, particularly you can observe in winter months here, or we can observe when you go to hill station, beautiful it will be. The leaves will have those drops. Don't confuse it with dew drops. That is different, like condensation of that water because of that atmospheric thing. That is dew drops. I am not talking about the dew drops. I am talking about the what you called the hyadothode, that is the guttation, where you see the exudation of those xylem sap drops, which will get accumulated, particularly sometime in the night as well as in the early in the morning. You can observe that. So hyadothode has to do with what? With guttation. And what about the xylem sap? We know that is with the ascent of sap. So the xylem thing, what? It is going to help in the upward movement of water and minerals, yes, which we call it as the ascent of sap with the xylem tissues. So this is the option. So what should be option? Yes, I see many people have answered it very right. So stomata with transpiration, hyadothode with guttation, root hair with absorption of water, and xylem vessel with the ascent of sap. So when the reason when we see students, why such easy questions you are making here, giving me all right answers, but what happens when the same scenario in the exam? The reason is because when you take up exams, there is something called the F factor. What is the F factor? The fear factor, the tension factor. Those are the one which will block your mind, and even the smaller ones which you know very well, you will not be able to solve it out. So that is why when you attend lot of rapid sessions and take up lot of revision tests and practice, that will make you very perfect in choosing out, so that you will be tension free. So you know that how to handle any of these are asked, so you can manage. So that is the significance, like how you have to take your biology test. Okay. Now here we are coming on to the other important question. Just previously we talked a lot about guttation. Here there is one thing, see, we are talking about the process of guttation takes place. Option A, when the root pressure is high and the rate of transpiration is low. When the root pressure is low and the rate of transpiration is high. When the root pressure equals the rate of transpiration. When the root pressure as well as the rate of transpiration are high. So they are asking about guttation, which I already told. It is through a specialized structures, like you can see in the leaf, that epidermal thing where you see the small pores like that will open up, and where you can see the exudation of drops. Okay, they are called the hydathodes. Achha. So what is happening here? So there is excess water, those droplets are formed from the hydathodes. They are like small pores like you see on the leaf margin. So what is the exact reason? See, whenever the root pressure is very high, okay, so and the root what you call the rate of transpiration is low, only in this circumstances the guttation will occur through. See, when root pressure is low and rate of transpiration is high, there is somewhere like where you see the water scarcity. So in these cases this cannot happen, or whenever it is equal also you won't have that exudation of water. Or when root pressure as well as the rate of transpiration is high, a point of equilibrium would be attained. So even here also you don't find the exudation. So what should be your answer? It should be the root pressure is high and the rate of transpiration. That's why you observe that particularly in the mid of the night as well as in the early in the morning, that exudase which comes out in the leaf margin. Okay, so that you have to know. Yes, the answer is C. So how beautiful, see those dew drops. They are not dew drops actually. So what you see, they are not dew drops. The dew drops are different, which are condensed from that atmospheric changes because on the plant surface. They are not that, but rather those exudases which come from that small tiny pore like opening on the leaf margin, which we call it as the hydathodes. Okay children, you are able to follow? Yes. So any question on guttation, it can be put forth in either of these ways. So you should be able to tackle it out and answer also. Okay. Okay. So we'll go to the next question. Yeah, I said already in this chapter we have a lot of diagrams with match the columns. So here we have another interesting match the column. They have given a lot of theories here and they have given a lot of scientists' name. So that is why it's important in this transport chapter to read lot of scientists' name, which is very important. So here a lot of theories are given. What are the theories? We have the relay pump theory, the transpiration cohesion theory, mass flow theory, pulsation theory. And here we have our honorable scientists, Dickson and Jolly, Sir J.C. Bose, Godlewski, Ernest Munch. So we are going to choose the correct or match the correct name and the theory of these people. Now when it comes to the relay pump theory, okay, so we are very well aware of this relay pump theory. So this was proposed by Godlewski, where he spoke about that vertical pump, the vertical pump of water through the plant, that ascent of sap, that was mentioned by the Godlewski, right? We studied about this. What about the transpiration cohesion theory? This transpiration cohesion theory, like they will be talking here about, they will say that the transpirational pull is there, that whatever that creates the water to get absorbed, and that is the reason for that ascent of sap. So this transpiration cohesion theory, this was actually proposed by Dickson and Jolly. Yes? Yes. Then what about the mass flow theory? This was proposed by our famous Ernest Munch, where he will talk about the movement of sap in phloem. So that is important. What about the pulsation theory? This was done by our Indian scientist, Honorable Sir J.C. Bose, which is also called the vital. This theory is also called what? Vital force theory. So even in the exam, they can twist this and ask. That's why I am emphasizing vital force theory. Acha. So what happens here? The inner cortical cells of the roots, they absorb the water from outside and pump the same into the xylem channel. So that is happening here in the pulsation theory. Okay, so this was proposed by whom? Sir J.C. Bose. So what should be your nearest answer? Yes, I see a lot of people, you have just given exact answers. Yes, you are all right. So this is regarding the name of the theories and the scientist who proposed it. Okay, very good children. Now coming to the next interesting question here, the proteins in the membrane responsible for facilitated diffusion and active transport show some characteristic like being highly selective, being liable to saturate, responding to inhibitor, being regulated by hormones. So we have to choose which of this is more characteristic to them. So before going into them, let me tell you like what is facilitated diffusion and active transport. So when it comes to facilitated diffusion, okay, facilitated diffusion, okay, so you see we have channels like this, okay. So what is going to happen here? So you see here in a facilitated diffusion, okay, and in active transport, let me show. So you have some proteins. What are they? They are nothing but those channels. They are nothing but the channels what we are talking. So here what will happen? There will be binding of the particular molecules. Okay, there will be binding of the particular molecules, and there will be a configurational change happening in these molecules which will push the molecules either inside or outside the cell. Okay. So here in active transport, here we require what is the energy, which is a uphill transport. I have mentioned. So what is uphill? Here you require a carrier protein as well as the energy is needed. But what about in facilitated diffusion? You need a carrier protein, but there is no need of the ATP. Okay, and here there is something they are liable to saturate. That is why you can see when you compare the simple diffusion with facilitated diffusion, okay, after some time what will happen? It will attain what is called the Vmax. This is with simple diffusion. So as long as the concentration of the substance is there, what will happen? The rate of diffusion will increase. It is linear. But here after some time, here they will attain a saturation in facilitated diffusion. Okay, what we call it as Vmax, because after which what will happen? There will be no carrier proteins to transport these substances. The saturation will be there. Okay, so that is the main thing. So and another thing, they are all regulated by hormones as well as they can be inhibited. Since they are channel proteins, like you have lot of inhibitors. Like we have says like sodium potassium pump, we have sodium potassium pump blockers, we have like that say for sodium pump blockers, potassium channel blockers, like that we have so many blockers, right? So these blockers can very well block. Like for potassium channel blockers, we have the tetra ethyl ammonium ions. Like that we have so many agents that can block any of these channels. Okay, we have Obain for the sodium potassium pump blocker also. Like that we have many such channel blockers. So they are respond to the inhibitors, and at the same time they are regulated by hormones. So when you look all these things, being highly selective, yes, they are selective. They are very much selective for the carrier. So only those specific carriers, they can just come and sit into that chair where they are going to make the conformational change, and they are liable to saturate. They are responding to the inhibitor. They are regulated by the hormones also. So when you take all these, all of these is the answer. Excellent, Tirisha, Akshaya and Balamurugan, you have given the right answer. So always you think in the mind, the difference between active and facilitated, one is with the energy. That is a main difference. So that you have to know. Here ATP is required, where ATP is not required in facilitated diffusion. But both will need the carrier protein. That point you have to know. Okay students, very good. So we will go to the other important nice question here. Go through the comparison of different transport process given below. So they have given lot of different transport process. So you have simple diffusion, we have facilitated diffusion and active transport. Here they have given some properties and they have given whether the options like yes or no. So we have to see which is the right one. See, simple diffusion, facilitated and active transport needs carrier protein. Everywhere it is SSS. Uphill transport, no SS. Transport saturates, no. Needs ATP, no SS. Now first we will start deriving at it. Okay. So simple diffusion, facilitated and active. Needs carrier protein. So I taught you already this needs a carrier protein, active transport needs a carrier protein, whereas here it does not. So that option A will not fit into it. What about uphill transport? I already said, okay, imagine a car is here. To push the car to the top of the hill, what you require, you need the energy, right? So when I say it is from a lower concentration to higher concentration is the uphill transport. Whereas passive transport is what? It is always from higher to lower. Okay, so that way you roam over one hill. So what here? In simple diffusion, no. In facilitated diffusion, yes. So when it comes to diffusion, it is a type of passive transport. So only this it will fit into it. What about the transport saturation? So transport saturation, I said here there is no saturation. As long as you have the concentration of substance, there depending on the rate of diffusion is linear. We say the concentration of the substance is directly proportional to the rate of diffusion in simple diffusion. But where about in facilitated and active? Yes, you have a point at which they will get saturated because they have the carrier protein. Okay. Even see, for example, we are going for some boating. Okay. So they say four people sit in a boat. You will be taken from one point to another point. So here we have people where we will be given token and we will all be waiting in a queue. And after some time, like what will happen? Like there will be only 4 boats at a time and others will wait. Same way that saturation will happen since there is a carrier required. The same thing is happening here also, that facilitated and active. Needs ATP, here it doesn't need ATP. Even in facilitated, no ATP. Only in active transport that ATP expenditure is happening. So what should be your correct? None of these. So none of these is the correct thing. So I see people giving the answers. Tirisha, Akshaya, Jeevita and Balamurugan. Very good. You have given the right answer. Excellent students. We'll go to the next. Go through the 4 statements given below. So they have given 4 statements and out of this I think the correct statements we have to choose. The root pressure provides a light push in the overall process of water transport. Okay, most plants meet their water need by transpiration pull. The greatest of root pressure may be to re-establish the continuous chain of water molecules in the xylem vessel, which often break under enormous tension created by transpiration. Guttation is the cause of transpiration pull. So out of all the 4 statements which I just read through, what is the correct statement? So when you look into the correct statement, root pressure provides a light push in the overall process of water transport. Yes. What about here? The plant meet their water need by what? The transpiration pull, which we are aware. And same, this is the greatest the root pressure. What will happen? Those chains of water molecules in the xylem vessel, they often broke into enormous tension that is created by the transpiration. But what about guttation? Guttation is not because of transpiration and pull. Guttation, when you see, it is not because of the thing. It is because of the root pressure. It is not because of the transpiration. So this is wrong. So what are the correct statements? These 3 are the correct statement, whereas this is the wrong one. Okay. So what you people are giving are the right answer. So excellent students. Next, here we are coming to another question. In a plant, chief sinks for mineral elements are apical and lateral meristems, young leaves. Third option, developing flowers, fruits, seeds, orange, that is storage organ, and all of these. So what we are going to see. So what do you mean by a place called sink? Sink is a place like wherever those products which has been assimilated or absorbed, they are going to be stored or used. So what are those places? So the growing regions of the plant, they are actually the area of sink. What is the source? The leaf where that is going to produce the products by photosynthesis, they are the source. The sink are the place where that is needed for the plant to assimilate and absorb or store. So like the growing regions of the plant. So what are the growing regions of the plant? The apical and the lateral meristems. Okay, we have those young leaves, the developing flowers, fruits, seeds, the storage organs. Okay, so all. So what happens? All of these is the answer. So yeah, all these are what? They are the sink. So all these are the places where the plant, the mineral thing, they are whatever the sucrose or whatever the products, the minerals, everything, they get stored up here or assimilated for the growing purpose. So these are the option. Okay, good. Now coming to the next question, the lower surface of leaf will have more number of stomata in a dorsiventral leaf, isobilateral leaf, both A and B, none of the above. See, when you have these options, see, dorsiventral leaf is characteristic of dicots, Isobilateral leaf is characteristic of monocots. Okay, so we will see what is happening. In a dorsiventral leaf, you see the stomata, actually you will have the distribution more on the lower surface than compared with the upper surface. So what is the reason? They want to prevent that water loss. What about in the isobilateral? There will be equal number of stomata on both the surface of the leaf. So that is very characteristic. So what is the option? It is the dorsiventral leaf. Yes students, we are right. Okay, next coming to this diagrammatic representation. See here they have given the diagrams as A, B, C, D and E, and they are asking us to label what are those paths. Okay, so here they are showing some process of transpiration, I hope. So it is a structure of a leaf. So what are that they are wanted to make it up. So here you can see that A, here they are talking here about the phloem. So what are these? Actually they are the xylem. Okay, so coming here on this epidermis, whatever you are seeing, okay, what are you seeing? You are seeing the epidermis, and you can see here beautifully the palisade layer. So what are you seeing here in the, here there is diffusion of that air, the air is getting escaped here through this process, and here in C you see these are the God cells, okay, and these are the stomatal pores. Okay, so what is that? So A is what? It is nothing but the phloem. Okay, and what you are seeing B is the diffusion of that air into the surrounding air thing, and C is the God cell. And what about the D? It is the xylem. Okay, and what about the E? It is the stomatal pore. And what about this? We are very sure it is the palisade, those long column like cells that is arranged, right? So that forms the palisade layer. Okay students, so they are asking us to identify what are the pictures, that is the label, and we have to make it out like this. Okay, very good. So next question here, in which method of transport in plasma membrane does not require carrier molecule? So in previous of the slides, enough of discussion we had with the transport of molecules. So they are asking which of this does not require a carrier molecule? Active transport, facilitated diffusion, simple diffusion, and sodium potassium pump. See, in active transport I already said it is nothing but it is a movement of molecules or ions from a region of lower concentration to higher concentration through a carrier protein, and it requires ATP. So this will not be our answer. What about facilitated diffusion? It is a type of passive transport where there is movement of molecule from region of higher concentration to lower concentration, and it is through a carrier protein. Okay, so this will also not fit into us. So what about the simple diffusion? Here it is the movement of molecule, okay, from a region of higher concentration to lower concentration. So that concentration gradient is the one which is making the movement of molecule. There is no energy, no carrier protein. So this will not, this would be the right answer. So no carrier molecule is required. What about the sodium potassium ATPase pump? It is an example of primary active transport. Okay, so where the carrier, where it requires a carrier protein as well as ATP. So these two things are must here. Okay, so you have given the right answer students. I see Tirisha, Akshaya, Balamurugan, Jeevita, excellent ma, you have given the right answers. Very good students, very good. So this is where you have to choose or derive the answer. Okay, good. Yes. We'll go to the next question. Stomata of a plant open due to influx of potassium ions, efflux of potassium ions, influx of hydrogen ions, influx of calcium ions. So I think you would have learned about this potassium theory, okay, that potassium ion pump theory that was proposed by Leavitt. Okay, so what does this theory actually they say about? So in this theory they will talk about that potassium ions that will be accumulated, that is actively absorbed. So that accumulation will decrease what? The water potential in the God cell. So that will become more turgid and it will make the stomata to open. Okay, so that is one of the reasons. So what should be your answer? It is nothing but the influx of potassium, because when potassium ions enter in here, when they get accumulated, what will happen? That water potential will decrease, so that the God cells The water potential, the guard cells will decrease, they will become more turgid, so that the stomata will be kept open. So that is the cause for the influx of what? The influx of the potassium ions. So you can also see that particularly how the stomata will be kept open, it will be happening in the daytime, right? So in the daytime, what will happen? The carbon dioxide concentration will decrease. So the malic acid, okay, the malic acid is formed in the guard cells. So now what will happen? This will dissociate into malate and hydrogen ion. Okay. So now what will happen? This is exchanged for the potassium ions in the subsidiary cells. Okay. So what will happen to the osmotic potential in the guard cell? It will increase. So what will happen because of that? There will be endosmosis of the guard cell. Okay. So that the guard cells will become more turgid, as I said earlier, and that will make the stomata to open. So these are the sequence of events that will happen. So the influx of potassium ions is responsible for the stomatal opening. Okay, that was one of the proposed theory given by Levitt. Okay. What is the correct order of decreasing imbibing capacity? What do you mean by imbibing? Something which has the capability to absorb that water. Agar, pectin, protein, starch and cellulose, or it is agar, protein, pectin, cellulose, starch, or it is protein, agar, peptin, starch, cellulose, agar, starch, protein, cellulose and pectin. So what should be the option? First, let us understand our cell wall, whatever you see, this is actually made up of colloidal substances, which we are very much aware. They are made up of cellulose, right? They are made up of pectins, they are made up of hemicellulose, right? So this we are aware of it. Okay. Now these substances, they are all colloidal and as well as they are hydrophobic in nature. That is, okay, so the imbibing of water is actually a physical process. So, okay, so what is happening, you can see it's like a special type of diffusion where there is movement of those water molecules which will be along the diffusion gradient. So you can see the plant imbibance, actually the agar is supposed to be the more potent. So what happens, which is in the order of decreasing, when you see the agar has the more, then comes in the order of decreasing, then comes the pectin, then comes the protein, then starch. So this is the order in which the imbibing capacity is placed in the plant. Okay students, you are able to follow? Yes. We'll move to the next. Yes, Trisha, she has given the right answer. Excellent ma. Akshaya also. Very good. We'll go to the next question. So increase in carbon dioxide concentration around leaf results in rapid opening of stomata, partial opening of, actually it should be partial closing of stomata, okay, complete closure of stomata. There will be no effect on stomatal opening. So what should be there whenever there is more carbon dioxide? Okay, so imagine there is going to be more carbon dioxide. So what is going to happen? When there is more carbon dioxide, what is happening? The asbestic acid, I hope you all know, that will get released. Okay, so there will be no potassium ion exchange. Okay, so what will happen in the guard cells? The guard cells, because there is no potassium, that will lose the turgidity. And so what will happen? The exosmosis will happen in the guard cells. Exosmosis will happen in the guard cells. Okay, so they will become more flaccid. So that will result in the partial closing of the stomata. Partial closing of the stomata. So that should be your answer. Okay, rather than the complete closure. Okay, so here as the concentration keeping on, we are talking about around the leaf, right? Okay, so as when it keeps on increasing, then only there will be that complete closure of the stomata. Okay, so what should be your answer? It should be, yes students, very good, you are giving the right answer. It should be the partial closing of the stomata. Excellent. Okay, here there is another interesting questions where they have given about the cell sap. So you see in cell A and cell B respectively, where they are talking about the solute potential, the water potential which they have given. So they have given separately in cell A and cell B, and we are going to find out the answer for this. Let me work out on it. See what happens here, they have given that it is in cell A, it is minus 900, okay, that pascals. What about the solute? Yes, it's but we know that already it is a what you call the addition of both these make up the solute potential. Okay, so that is how it comes. So when you make it up, it is nearly coming out to the 300, because what do you say, minus 900 plus, okay, plus 1200 comes to plus 300. Okay, so this is for the cell A. But what about for cell B? Okay, I have worked out in the next slide, like you can see here. So what happens here, it is minus 600. What about the solute? It is minus 1000. So when you are going to add, when you are going to subtract both, okay, so when you are going to subtract both, how much you are getting? You are getting 400. So what should be your answer? It is very good, students. Akshaya, Trisha, Balamurugan, excellent. You have given the right answer. What about the potential that is there in the cells? Excellent. Okay, here comes the next interesting question. During sodium potassium pump, 3 sodium and 2 potassium are transported, 1 sodium and 2 potassium are transported, 3 sodium and 3 potassium are transported. It depends on the requirement of cell. So what your answer should be? I am waiting for your answer. Yes. Yes, very good, students. You are giving the right answer. So what happens in sodium potassium pump, which I have already explained? It's a type of primary active transport, right, where it requires ATP, it requires a carrier protein. Okay, this sodium potassium pump is operating in all the cells of our human body. That is one clue which I say. And Obain is a drug that can block the sodium potassium pump. These are all additional points which I am giving. Let me teach you how the sodium potassium pump. So you have a pump like that. Okay, I'm sorry. You have a pump like that. So what happens here? You have the areas where it can bind with. So what happens? Usually 3 sodium and 2 potassium. So usually the 3 sodium ions, they will be pumped out for every 2 potassium ions that is pumped inside the cell. Okay, so this is quite characteristic. You can observe in the neuron. That is the one which is creating the difference in this electrical potential and initiating the development of the depolarization. Then the wave of depolarization is traveling as a action potential. That is what. So you can see that that is the reason for the sodium ions to be more outside than inside. Okay, so 3 sodium ions will be pumped out for every 2 potassium ion pumped in. That one deficit of that ion, okay, whatever that is pumped out, and 2 potassium, that is creating a what? That imbalance, right? Because 3 is going out for every 2 entering in. So that difference is also created. Okay, so this you have to know. So the option would be what? 3 sodium ions and 2 potassium ions are transported. Now according to the potassium pump theory of Levitt, which we already discussed in previous slide, the starch is incompletely oxidized to PEP during night. Malic acid dissociates into malate ion and protons in the subsidiary cells. Movement of hydrogen ions from guard cell to subsidiary cell is active. Potassium malate decreases the osmotic potential of guard cells during day. So what should be your nearest answer for this? We already spoke about the what you called the potassium pump theory. So what should be your option? Yes, I am looking in your answer. Okay, so we saw about during night time how when carbon dioxide concentration, that is during the daytime, when it decreases, what all is happening? We saw that malic acid in the guard cell dissociating itself into malate. Yes, we have the hydrogen ion, how it is exchanged for potassium ion in the subsidiary cell, and then we saw that entry of this potassium ion is making it more endosmosis in the guard cell, making more turgid. The stomata is closed, that is the stomata is open. What about here? Here the carbon dioxide is increased during the night time. What happens? Aspartic acid is released. No more potassium exchange, it's all stopped. So there will be exosmosis of the guard cells. So they become more flaccid. The guard cells become more flaccid, and so the stomata will be kept closed. Okay, so this is the scenario that we spoke. So what is happening? The starch is incompletely oxidized to phosphoenol pyruvate during night. So this cannot be the one, right? Malic acid dissociates into malate ion and protons in the subsidiary cells. Okay, this is not. But what is happening actually? The movement of hydrogen ion from guard cell to subsidiary cell is active. The potassium malate decreases the osmotic potential. It's not decrease, it will actually increase the osmotic potential of guard cells during the day. That is why they become more turgid. Okay, so what should be your answer? The starch is incompletely organized to PEP during the day, not the night. Malic acid will dissociate into malate ion and proton in the guard cell, children, not the subsidiary cell. And at the same time, the potassium malate will increase in the guard cell during the day, not decrease. Okay, so that should be your answer. That is why I am telling you, when you are choosing it, you should see the slightest thing, the subsidiary cell and guard cell, which is quite near, but where is that exactly that is happening? That is where matters it. Potassium ion is with the subsidiary, but actually the malic acid, those dissociation will happen in the guard cell, where the chloroplasts are there. Okay, so that concepts, key concepts you should know. Okay, very good. Akshaya, Jivita, Tirisha and Balamurugan, you have given the right answer. Yes, good. Now, purple cabbage leaves do not pass out color in cold water, but do so in hot water because hot water enters the cells faster. Pigment is not soluble in cold water. Hot water destroys cell walls. Hot water kills plasma lemma and makes it permeable. So, you take a purple cabbage, you are just washing it in a cold water, nothing happens. But when you do so in a hot water, the color slightly changes. So what is the reason? So the reason is because what? When you make it with a hot water, it will kill the plasma lemma. So cell membrane permeability will be altered because of the temperature change. We know that what are the things that will alter the permeability of the cell membrane. One such is the temperature. So once when the hot water changes the permeability, what will happen? It will kill the plasma lemma, and that is the reason. Okay, so the answer is what? The answer is hot water, it kills the plasma lemma so that it is making it more permeable. They don't destroy cell wall. It is not like making them not viable. Okay, the pigment is not soluble or hot water enter it faster, nothing like that. It is just altering the permeability, and so some amount of color is getting lost. So that is what is the answer. Okay, very good students. Now you see there is a botanist who discovered a mutant plant that was unable to produce materials that form the Casparian strips. Okay, this plant would be. So what would be? So there is a mutant he made. So there was no Casparian strip. So how it would be? Unable to transport water or solutes to the leaves, unable to use its sugar as a sugar sink, able to exert greater root pressure than the normal plant, unable to control amounts of water and solutes it absorbs. So what is the role of Casparian strip? So we saw that Casparian strip which is there present on the endodermis. The main role is what? To control the water and solute absorption, which we have already studied that in the apoplast symplast pathway. So here these are all the job of what? The xylem, okay, or you can see the role of a phloem. So what should be your answer? It should be, yes, you saw the apoplastic root and this is the symplastic root. Okay, so what you see is what? The Casparian strips, right, which is present in the endodermis. Okay, so what they are going to check the entry of water and solute inside the cell, inside the xylem vessels. So that is the thing. So what should be your answer? It is unable to control the amount of water and solute. So since you imagine some mutation has happened and there is no Casparian strip, just like that water and solute. So you don't have a checkpoint entry at all. So just like that they can enter. So that is where you have to make it up. Very good students. Then we have another interesting one. If flowers are cut and dipped in dilute sodium chloride solution, then transpiration is low, endosmosis occurs, no bacterial growth occurs, absorption of solute inside flower cell take place. So you are taking a flower, you are cutting it, okay, and you are dipping it in a dilute NCl, sodium chloride solution. So what all things will happen? So what all can happen? The endosmosis will happen because you are having, what is endosmosis? Endosmosis is the movement of the solvent Okay, there is movement of solvent which is going to be from lower solute to higher solute concentration. Okay, so when you are dipping it like that in a dilute, what will happen? All that it will move from the dilute NaCl, the solvent from there will move inside the flour, so that endosmosis will occur. So that is the reason. Okay, very good children. Not like bacterial growth or absorption of solute or transpiration, nothing to do with it. So it's quite logic here. Next, coming here to another question, the root cap has no role in water absorption because it has no direct connection with the vascular system, loosely arranged cells, no cells containing chloroplast, no root hairs. So something with water absorption, this chloroplast that is not going to make, or loosely arranged cells are not there, or with the vascular system. So when you take root cap, it's one of the structure that is enclosing the root. Okay, but what is missing there? For absorption of water, we need what is called the root hair. Since the root cap is devoid of root hair, water absorption will be having no role there. So what should be your answer? No root hairs. So that should be your option. Okay, so the answer is what? There is no root hairs in the root cap, so it has got no role in the water absorption. Okay students, yes. So the next, coming to the last question, you are going to find out which of the following statement are not incorrect. Or not incorrect means what? Some statements are going to be true. Okay, water and minerals and food are generally moved by mass or bulk flow system. Bulk flow can be achieved either, okay, through a positive hydrostatic pressure gradient and a negative hydrostatic pressure gradient. The bulk movement of a substance through the conducting tissues of plants is called translocation. The xylem translocates organic and inorganic solutes mainly from roots to the aerial parts of the plants. Phloem translocates water, mineral salts, some organic nitrogen and hormones from leaves to other parts of the plants. So among this, some are not incorrect. Find out the option. So the answer would be 4 and 5 only. They are not incorrect. Okay, so these two are not incorrect. Okay, because xylem translocates, right, that organic and inorganic solutes. So where is the xylem that translocates? It translocates what? It translocates the minerals and water, ascent of sap. What about phloem? It translocates it acts as a source, no, right, from the particularly wherever that is manufactured, from there it is going to go move it or push it to the area of sink. Okay, so those two are incorrect. So that you have to know. So I think students, today with a rapid revision round on transport in plants, especially Balamurugan, Akshaya, Tirisha, Jeevita, Murugan, okay, you all had a very nice interactive, because I see before I post the questions, you people were just giving up answers, and it was very interactive and very informational. So it was a very nice learning session. So I hope we will meet you in the next rapid revision rounds on another set of interesting topics. Okay, so waiting you all to meet you soon. Thank you students for being patient and interacting very well. A big hand of applause. Thank you students.